Good morning, one and all. I welcome you personally to my webinar on Importance of Fermentation and Bioprocessing. Here I am Arvind, faculty from Sri Dharmasthala Manjunatheshwara Postgraduate Center, Department of Postgraduate Studies and Research in Biotechnology. I welcome. So today's topic, the importance of why fermentation as an upstream is important and the basics of downstream processing. So if we glance through the production cycle of what many major biopharmaceutical companies in the world go through, is the first stage is upstream processing. So here the genetic engineering of microorganisms is done, and it follows either the microbial fermentation or the cell culture fermentation. Then it goes for the next step, downstream processing, that is isolation and purification of an active compound or a protein from it. Then you set up the formulation where the final DP, that is a drug product, is obtained from the drug substance. So coming to the definition, what is fermentation? Fermentation is the term used by microbiologists to describe any process for the production of a product by means of mass culturing of microorganism, that is a particular microorganism. So it is the controlled way how we deduce an important product from the microorganism by mass producing the microorganism, that is in large. So coming to the basics of fermentation, the product can either be the cell which the microorganism produces, the cell itself, it is referred to as the biomass production, or the metabolite produced by a microorganism, which is referred to as a product from a natural strain. A microorganism's foreign product is a third part, that is, it is referred to as a product from the recombinant DNA technology, or genetically modifying or genetically engineering that strain. That is, we get a recombinant strain and it produces a different product that is called as a foreign product. So coming to fermentation, here we will go through what is a batch fermentation. So a batch fermentation is a closed type of fermentation. It is a closed system at time t equals to the initial time 0. The sterilized nutrient solution in the fermenter is inoculated with the microorganisms and incubation is allowed to proceed. So in the course of the entire fermentation, nothing is added here. So only dissolved oxygen levels are maintained. So when if it is the fermentation, if it is an aerobic fermentation, and the acid and the base levels to control the pH is. So the batch fermentation, here the composition of the culture medium, the biomass concentration, the metabolite concentration generally change constantly as a result of the metabolism of the cells. So whatever media we have added into the bioreactor or the fermentation to occur as a batch. So here the biomass concentration keeps on varying, the metabolite concentration also keeps on varying. So after the inoculation of a sterile nutrient solution with a particular load of microorganisms and a pure culture of microorganisms, so cultivation under important physiological conditions, we see the growth of microorganisms in different phases. So as you can see in the picture, if you can plot a graph versus time, the cell count, so the microorganism growth phase occurs in 4 different stages, that is the lag phase, log phase, stationary phase and the death phase. So coming to the growth phases, So before that, you can log into my channel, you can subscribe my channel Aravinda Narayana on YouTube for many more such webinars in the days to come. And here this is a part of encouragement video for the students who will take up this bicep course that is under the domain fermentation and bioprocessing. So coming to the topic, the lag phase, physico-chemical equilibration between the microorganism and the environment is happening at the initial phase, that is the log phase. The second stage is the log phase. Growth of the cell mass can now be described quantitatively. So doubling time occurs, doubling of cell number occurs. So if there is one cell, then doubling occurs. It occurs 2, 4, 8, 16, 32, such as 2 to the power. So next is the third phase, that is the stationary phase. As soon as the substrate is metabolized or toxic substances are being accumulating in the fermenter, the growth of the microorganism in the media slows down or is coming to an halt. That is the stationary phase. And in the death phase, all the media components, the energy reserves of the cells are exhausted and it goes into the phase. So this all comprises the upstream part. Coming to the downstream processing, we need to know the basics of what is done in an upstream and what is done in a downstream, which will form the basis or the foundation step to learn more, to acquire the knowledge in something called as a domain area of fermentation and bioprocessing, which is a skill development course sponsored by the BICEP, that is the Biotechnology Skill Enhancement Program, sponsored by the Government of Karnataka under the K-Tech governing body. So coming to downstream processing, it comprises all operations required for the extraction and purification of a particular product produced by the fermentation process, such as it may be a biotechnological process where you employ microbial fermentation or other tissue culture or transgenic type of producing plants and animals. So mainly we are focusing on fermenter level only because we are extracting or getting the product from a microorganism where we utilize the bioreactor. So here it can undergo different phases of processing, that is either cell separation or cell disruption. Cell separation, where the clarified culture medium will go into the next stages of product recovery, concentration, polishing, formulation, and if it is cell disruption methods also, cell debris are removed and it goes into the successive steps. So if you see in totality, this is the picture how a downstream processing occurs from the fermentation broth. Once the media is clarified, the cell disintegration can occur because the product can move from the microbial cell as an inclusion body, or the product can remain soluble in nature, or the product membrane can get associated. So in all the three different steps, how the product is in the microbial cell, out of the cell, or associated with the cell, here we have to go to different kinds of downstream methods. That is downstream because it is after the fermentation. That's why the purification part. So this is a general flow chart of purification of a protein from a culture broth. So to understand this, we need to have a thumb rule in our mind. It may not always be necessary to follow a long and difficult path to obtain a pure protein, because proper planning and a smart choice or integration of different steps can lead to a cost-effective, efficient process. So we have to look into the economic aspects of downstream processing also, because recombinant technology has established well in upstream processing, but for the biopharmaceutical industry, downstream processing or this bioseparation is a very challenging task, because 80% of the product costs are incurred in the downstream process. So if you see the biosuppression costs or the downstream costs as per various biotechnology companies working in food under the food additives, nutraceuticals, industrial enzymes, diagnostic proteins or therapeutic proteins, it is valuated as per you see in the. So coming, why you need to have downstream purification? Because you need to reduce the bulk into minor component, enrich into and concentrate it into a particular product, remove specific impurities from the product, prevention of catalysis of protein and preventing your protein from getting lost, recommended product specifications, because there are various requirements, guidelines. As per the guidelines you need to do the product purification. And next, enhancement of protein stability and reduction of protein degradation. The product will get degraded. That is, all these forms the essential components of a downstream process. So you have to modify the upstream process in such a way that it aids in downstream purification. So selection of organisms that do not produce undesirable pigments or metabolites, modify the fermentation conditions, think of a precise timing of harvesting from the fermentation, then control of pH and temperature after harvesting, addition of other flocculating agents and addition of antifoams which should not interfere with the purification process. So the protein products, what are the important products produced because of the complete process of fermentation and bioprocessing? You can see it ranges from food to industrial enzymes to diagnostic enzymes to pharmaceuticals. So most of the downstream processing industries follow this RIPP method, that is removal, isolation, purification and polishing. So removal of particulates, that is the first common step in the operation sequence of a downstream process, where it involves the primary basic level of purification employing the method of filtration, centrifugation, either maybe settling, decanting also. Second step is the primary isolation, where you use ultrafiltration steps to increase the product concentration. Next you go for the main purification step, where you use several types of chromatography related methods, and the final product isolation, where a formulation is done, that is by using freeze drying or other stability considerations you need to think of for final product isolation. So the unit operations in downstream processing are what I have shown you till now and what I am showing it in the slide. It consists of this in totality, from cell separation, cell disruption, clarification, concentration, high resolution techniques such as chromatography, electrophoresis, dialysis, and final packaging, that is the finishing or the packaging step by crystallization or filtration. So coming to the end of the complete basics of fermentation and processing, you can opt to choose this as a skill development course at Shri Dharmasthala Manjunatheshwara College, where we have a separate postgraduate center and we also have an exclusive 2000 square feet lab to cater to the needs of the student who wants to study and develop and become professionals in this bicep course. So thank you. Keep watching my webinars. You can log on to my YouTube channel, subscribe the channel and share the channel. Stay safe. Take care. See you.